Good morning, everybody. Thank you for your introduction. I am a surgical resident in the University Hospital of Nantes, and I'm very pleased to present that topic here. I have no disclosure. As you should know, the colorectal cancer is the third most frequent cancer in the world, with 40% of metastatic forms. The overall survival of metastatic cancer is about 11%, attesting that the pathophysiological mechanisms underlying the colorectal cancer development and dissemination are not completely understood. Recently, the tumor microenvironment has been identified to play a crucial role in the cancer progression and dissemination. This tumor microenvironment is composed of the cells resident or recruited by the tumor surrounding it. And in fact, there are bidirectional interaction between the tumor cells and the cells of the TME. The tumor cells can alter the properties of the cells located in the tumor microenvironment, and conversely, the cells of the TME can promote the tumor cells' proliferation and dissemination. And inside this tumor microenvironment, the role of the enteric nervous system has not been well studied so far. And as everybody knows here, I hope, in the colon and in the rectal wall, there is a specific intrinsic nervous system called enteric nervous system and composed with enteric neurons and enteric glial cells, which communicates with intestinal epithelial cells through the basement membranes. And this enteric nervous system controls some epithelial barrier properties, and conversely, the epithelial cells favor the neuronal survival via the nerve growth factor, NGF. And we don't know if such communications persist in tumoral condition, but what we know is that the phenomenon of perineural invasion, which are defined as tumor cells surrounding the neural structure of the enteric nervous system, have a negative impact on the global survival rate in the colorectal cancer. But whereas close contact between tumor cells and the myenteric plexus have been suspected, the exact pathogenesis of the perineural invasion remains unclear. And in addition, some studies identified that in the colorectal cancer microenvironment, there was an increased neurite density. As you can see here, I'm sorry for that, but you may not see it clearly, but there was an increased number of neurite in the colorectal cancer sample as compared to the healthy colon. And moreover, Ayakawa and colleague identified an overexpression of the neuronal growth factor by colorectal cancer cells and by colonic epithelial cells, which are responsible of an increased nerve density in the submucosal plexus. So, based on this data, we hypothesized that colorectal cancer cells exert a neurotrophic effect on the enteric nervous system, and that nerve growth factor is involved in this neurotrophic effect. And in addition to the increased neurite density that I presented you 2 slides before, we could identify that there were also an increased neuronal density. As you can see, in the tumor, the number of neurons per ganglia were superior than in a healthy area, and this was true in both myenteric and submucosal plexus. And interestingly, we could identify the same phenomenon of increased neuronal density in a chemically induced colorectal cancer mice model. To assess the effect of the colorectal cancer supernatant, healthy and colorectal cancer supernatants were added to primary cultures of enteric nervous system and enteric neurons. Then the co-cultures were immunostained using an anti-HU antibody to stain the neuronal bodies, and an anti-TUJ antibody to stain the neurites. The cultures were then observed using standard microscopy, and finally, the number of neurons per ganglia and the neurite length were counted and analyzed. First, what we can see is that the addition of tumor supernatant in the cultures was responsible of an increase of the neuronal density. The number of neurons per ganglia was superior in tumoral condition than in healthy condition, and also the neurite lengths were increased by the addition of tumor supernatant in the cultures, suggesting that the colorectal cancer supernatant has a neurotrophic effect on the enteric nervous system. This is the first point. And in addition, we could see the same effects of tumoral epithelial cells from CACO2 cells supernatant in the cultures concerning neuronal density. The addition of tumor epithelial cells supernatant increased the neuronal density in the culture, suggesting that this supernatant from CACO2 cells contains neurotrophic factors. Then, to assess the involvement of the neuronal growth factor, we first add human NGF on the primary culture of enteric nervous system, and on the other end, the NGF was blocked using an anti-NGF blocking antibody in the cultures containing healthy supernatant or tumor supernatant. The addition of NGF in the cultures was responsible of an increase of the neuronal density, as you can see, in a concentration-dependent manner. So, not surprisingly, we can say that the neuronal growth factor has a neurotrophic effect on the enteric nervous system. Then, let's see the effect of blocking NGF in the cultures. As you can see, the neurotrophic effect of the tumor supernatant was not significant anymore after the inhibition of the neuronal growth factor in the cultures, suggesting that the inhibition of NGF decreases neuronal density in the enteric nervous system. So, Chairman, ladies and gentlemen, as a conclusion, we can say that colorectal cancer microenvironment contains a soluble factor which is able to induce an increased neuronal density. This mechanism could be mediated via a neuronal growth factor dependent pathway, as we can see. But if real neurogenesis occurs, has to be confirmed. For example, using a model of lineage tracing of the neuroglial precursors in a model of chemically induced colorectal cancer in mice. Thank you for your attention.